Good morning students, Ms. Poonam this side to continue chapter 2 sexual reproduction in flowering plants. In today's class we will be talking about the pollination, pollinator agents, outbreeding devices, and then finally we will talk about the pollen pistil interactions. So let's start today's class. We will start with the pollination. So pollination, what exactly the pollination is? Pollination is the transfer of pollen grain from male gamete to the female gamete, or you can say that from anther to the stigma. And pollination is of two types, self-pollination and the cross-pollination. And if we'll talk about the self-pollination is further categorized into three main categories: autogamy, genetogamy, and the xenogamy. So first we will be talking about the autogamy. Autogamy is the transfer of pollen grain from anther to the stigma on the same flower. Means in the same flower, the male and female gametes are present. Here the two types of flower can be present, clastogamous or the chesmogamous. What happened? What's the difference between these two terms? Clastogamous means flowers which do not open, means the flower which are closed one. The male and female part are not exposed. Clastogamous flowers are autogamous as there is no chance of cross pollen lending to the stigma. So as there is no chance of cross pollination, means no need of the pollinators. So here the examples can be viola and the oxalis. And the second types of flower can be chesmogamous. Chesmogamous means male and female, means anther and stigma both are exposed. If both are exposed, means there is a 100% chances of the cross pollination. So these type of flower can come under the autogamy. So moving to the next one, geitonogamy. It is the transfer of pollen grains from anther to the stigma to different flowers of the same plant. सेम प्लांट मीन्स एक ही प्लांट के दो डिफरेंट फ्लावर है और वहाँ पे जिस टाइप का क्रॉस पॉलिनेशन है दैट इज कॉल्ड द geitonogamy. And the next type is the xenogamy. Xenogamy means transfer of pollen grain from anther to the stigma of different plants of flower of the same species. मतलब एक ही species के दो different plants है और दो different flowers के बीच में जो cross pollination हो रहा है that's come under the xenogamy. So you can see the picture on the slide. Here the genetogamy can be taken place by the self pollination and the cross pollination, and the xenogamy it is taken place by the cross pollination. So moving to the next one. So our next topic is agents of pollination. The agents of pollination may include the abiotic factor and the biotic factor. Abiotic factor can either be the water or wind, and the biotic factor can be insects, butterfly, honeybees, etc. Actually what happened, the large number of pollen grains are produced by the plants using the abiotic mode because there is a chances of destroying of the large number of pollen grains, so that it is a natural process. The large number of pollen grains are produced by plants. At least few of the pollen grains can reach to the female reproductive part during the pollination process. So here we will have a look on the adaptation in flowers for the pollination. If we will talk about the wind pollination, in the wind pollination the pollen grain will be light, non-sticky and the winged. And anther will be well exposed, stigma will be large and feathery, flower will be one ovule and arranged as a inflorescence. An example can be cob, cotton and the date palm. If we talk about the water pollination, it can be carried out in bryophytes, pteridophytes and the algae. In this, pollen grains will be protected by the mucilaginous covering. An example can be freshwater plants like the Vallisneria, Hydrea, seagrass, Jostrera. And the main feature of the wind and water pollinated plants will be they both type of pollinations, they produce the pollen grains in large number and do not produce the nectar. Our next type of pollinator is insect pollination. Under this, the flower will be large, colorful, fragrant, rich in nectar. Because if the flower will be very colorful, then only the insect will automatically, they will attract to the flower. Under that, pollen grains will be quite sticky and stigma also will be sticky, and the certain rewards of pollinators will be the nectar and edible pollen grains, provide safe place for the laying eggs. Example can be Amorphous and the yucca. These both are the examples comes under the insect pollination. Our next topic is our outbreeding devices. What exactly the outbreeding devices? The breeding devices can either be the inbreeding devices and the outbreeding devices. If we talk about the outbreeding devices, it discourages the self-pollination and it encourages the cross-pollinations, as the continuous self-pollination leads to inbreeding depression. Because if inbreeding will be there, then definitely self-pollination will be carried out, and if the outbreeding devices will be there, then in that case the cross pollination will be carried out, because the pollen grain will be come from the other flower, and under this the pollen released and stigma receptively not synchronized. An anther and stigma are placed at different position. Mainly the unisexual flowers exhibit the outbreeding devices, because in the unisexual flowers only one reproductive part will be there, either male or female. So now moving to the last topic of the day, that is pollen pistil interaction. What exactly? The pistil has ability to recognize the compatible pollen to initiate post pollination events that leads to the fertilization. Means as the anther release the pollen grains to the female reproductive part, then finally that pollen grain has met to the ovary. And how does it take place? The pollen grain produce pollen tube through the germ pores to facilitate transfer of pollen gamete to the embryo sac. So it is the way to reach the pollen grains to the pistil part. Pistil part, under that it has to meet physically to the ovary that is present in the pistil, means that is present in the female reproductive part, and finally the both gamete meet and lead to the formation of embryo sac. So with this we have done with the pollination, and our next topic will be the double fertilization that will be done in the next class. I hope I made this topic clear to you, but if you have any kind of query, you can write down in the comment section. So thank you so much for being with me. For the further lecture, you can subscribe the YouTube channel Biology Classes with Miss Poonam DHALA. Thank you so much. Take care.